Thank you, everyone, for coming to our session, which is expanding the promise of gene therapy. I had the honor of introducing our panel, the members, the speaker to the stage. Actually, it's a great panel list of speakers that really requires no introduction. I mean, perhaps the only person that requires introduction on this stage is me. So my name is Audrey Chang. I am the Executive CMC Director at WuXi AppTec, and I have the honor of introducing. So the first speaker is going to be Dr. Jorge Santiago-Ortiz. And real quickly, I'm going to breeze through these introductions because I know everybody wants to get to the talk. But Dr. Jorge Santiago-Ortiz is a Senior Director of CMC and Controls at Apertura Gene Therapy. He's a chemical engineer with a background in process engineering, molecular biology, biochemistry, focusing, of course, on gene therapy and process development. So today, we're going to hear the latest with his talk, Beyond Barriers for Expanding Gene Therapy, with the intravenous AAV capsid for neurological disorders. Okay.
Wonderful. Great. Thank you so much, Audrey, for the introduction, and good afternoon, everyone. It's a pleasure to be here. I am excited to tell you more about the advancements that Apertura Gene Therapy has been making towards expanding patient access to gene therapies. I would like to start by telling you more about Apertura Gene Therapy. We are a New York City-based company that is engineering novel AAV vectors. We were founded in 2021 by Deerfield Management based on platforms that were in-licensed from the Broad Institute and Harvard Medical School. Our proprietary receptor-targeted capsid engineering platform allows us to engineer AAV vectors that specifically bind to human receptors of interest, therefore increasing the likelihood of clinical success. We have applied this approach to engineer novel AAV vectors that bind to the human transferrin receptor. These vectors are capable of efficient crossing of the blood-brain barrier, leading to widespread CNS biodistribution, as can be seen on the image on the right. Our platform relies on first identifying tissues and cells of interest, and then using a mechanistic understanding of what are the receptors that we can leverage to access these cells and tissues. We then engineer AAV vectors that can specifically bind to these human receptors, and also use humanized animal models that express the human version of these receptors to increase the specificity and the translatability of these capsids. Our platform is tissue and receptor agnostic, and we're able to develop novel AAV vectors under fast timelines. Furthermore, our engine co-optimizes manufacturability on top of the functions we're interested in, such as binding to a specific receptor, so that we can always ensure that these vectors can be manufactured. This technology has been pioneered by our scientific founder, Dr. Ben Deverman at the Broad Institute, over the last 10 years, and it is this deep level of expertise that makes us believe that we are the leaders in receptor-targeted approaches for capsid engineering. Now, as you may know, the field of AAV gene therapy has experienced a series of challenges that have been difficult to address using traditional capsid engineering efforts. We have capsids that have poor selectivity and poor distribution towards target tissues, especially in the central nervous system or the CNS. This can result in a requirement for high doses in order to achieve a therapeutic effect. We have capsids with low accessibility, given that they may be neutralized by the presence of neutralizing antibodies against AAVs, which a large fraction of the population has as a result of natural AAV infections. Capsids may have low translatability based on how they were developed and the animal models that were used to develop them. This includes non-human primates, which may not always translate well into the clinic. Lastly, engineered capsids may suffer from lower production yields and low manufacturability. We may also think about these challenges as opportunities to engineer better AAV vectors. For example, we can engineer vectors with improved tropism and improved selectivity towards the tissues of interest while detargeting them away from other tissues. We can also develop capsids with enhanced accessibility that are able to evade neutralization by pre-existing antibodies, thereby expanding patient access to gene therapies. We can develop vectors using a mechanism-first approach so that we can increase the translatability as we move from cells to humanized animal models to finally human patients. And lastly, we can engineer them to have higher manufacturability so that they can be compatible with existing manufacturing processes. Today, I will tell you more about how we have leveraged our capsid engineering platform towards the development of better AAV vectors and those that can improve accessibility to gene therapies. About 50% of the population has pre-existing antibodies against AAV as a result of natural AAV infections. These antibodies can render gene therapies inefficacious, and as a result, patients must be pre-screened for the presence of neutralizing antibodies before they can be eligible to receive gene therapies or to enroll in clinical trials. Therefore, it's really important that we develop vectors that can evade these neutralizing antibodies so that we can expand the access of these gene therapies to more patients. This is particularly important when we're thinking about indications that have adult populations as patients, given that these older patients are more likely to have developed neutralizing antibodies throughout the course of their lifetime. This is an illustration of our capsid engineering platform. Our approach starts with identifying a receptor of interest, generating diverse AAV capsid libraries, and then subjecting them through a series of in vitro screening efforts so that we can isolate variants that can bind to the receptors of interest. We can then further diversify these libraries and subject them to additional rounds of in vitro screening as well as in vivo screening using humanized animal models before we're able to select lead candidates. As I mentioned, the first step in our capsid engineering platform is the identification of a receptor. At Apertura Gene Therapy, one of the tissues of interest is the central nervous system. So we have been working with the human transferrin receptor. This receptor is expressed on the vasculature of the blood-brain barrier and is able to shuttle AAV particles that bind to it. Across the blood-brain barrier through a process called receptor-mediated transcytosis. Because these receptors are expressed on the vasculature of the BBB, we're able to leverage intravenous administration of AAV capsids. And IV administration is a minimally invasive and not complex procedure that can also be supportive of expanding patient access to gene therapies globally. We have thus used our platform to engineer novel AAV vectors that can bind to human transferrin receptor and that offer best-in-class blood-brain barrier crossing capabilities. As you may have seen in our science publication last year, the TfR1 capsids are able to overcome many of the limitations of AAV9. To start out, these capsids can be administered intravenously, and they're able to achieve broad distribution towards the central nervous system, as can be appreciated in this image here, where green dots indicate successful cell transductions as measured by in situ hybridization of AAV episomes. These capsids, due to their higher gene delivery efficiency, may also be administered at lower doses. We can also detarget them away from tissues such as the liver. And finally, because we have been developing them with manufacturability in mind, these capsids are compatible with existing manufacturing processes for AAV, so their manufacturability is comparable to that of AAV9. Given that these vectors are capable of broad CNS distribution, we can leverage them towards the usage of in indications that are able to have multiple, we can leverage them towards some indications that are either rare or those that have large patient populations. So these can include Parkinson's disease as well as Alzheimer's disease, which can have patient prevalences on the order of hundreds of thousands of patients to millions of patients in the US alone. Many of these indications have adult patients, and as I mentioned, these are more likely to have developed neutralizing antibodies against AAV. So all of these points highlight the importance of delivery, efficacy, manufacturability, route of administration, as well as the ability to evade neutralization by pre-existing antibodies towards expanding patient access to these gene therapies. Apertura's TfR1 capsids have been engineered with accessibility in mind. Our antibody evasion platform confers mutations that enable these capsids to evade neutralization by pre-existing antibodies. these modifications are done such that manufacturability is retained, so we're still able to use commercially available resins to purify them. More importantly, these modifications do not come into conflict with modifications that are involved in receptor binding and transduction properties. So these modifications do not affect their tropism, and they are compatible not only with TfR1 capsids, but also with other families of engineered capsids. In collaboration with our scientific founder, Dr. Ben Deverman, we have established an in vitro transduction assay that enables the detection of existing antibodies against AAV in individual patient serum samples. Right now, I am showing you data for 59 individual patient samples of diverse racial backgrounds that have been collected throughout the US. And we can use this assay to understand whether these serum samples can be cataloged as having neutralizing activity against AAV9 or whether they do not neutralize activity. Furthermore, we can use samples that have been found to have neutralizing activity against AAV9 to assess the ability of capsids to evade neutralization by these antibodies. So I will now walk you through this data, which is a pretty complex figure, but please bear with me. On the left, we have a heat map where each column represents an individual patient serum sample that has been found to have neutralizing activity against AAV9. And each row represents an evaluation of a given AAV capsid and its ability to evade neutralization by antibodies. The color represents normalized transduction or transduction in the presence of serum over transduction without serum, expressed as a percentage. The purple, we have no transduction in the presence of serum, so this indicates that a capsid has been fully neutralized. And in yellow, we have 100% transduction, which means that it has been able to fully evade neutralization by antibodies. On the top, we have AAV9. And as I showed you on the earlier picture, given that these patient samples have been found to have neutralizing activity against AAV9, this data further confirms that. Then we have TfR1 CapX right here, which is our lead candidate TfR1 binding capsid, which shows some slight improvement over AAV9. On the bottom, we have TfR1 capsids that have been specifically engineered to have evasion properties against neutralizing antibodies. And as you can see from the high presence of yellow, these capsids are much more efficient at evading neutralization from these antibodies and are able to retain their transduction capabilities. Importantly, these modifications do not affect tropism, and this is what I'm showing you on the right, where we have images of M-scarlet, a fluorescent protein, delivered intravenously into mice that have been humanized to express the human version of the transferrin receptor. On the left, we have delivery using TfR1 CapX, and on the right, we have delivery with TfR1 CapXC, which is an evasive version of TfR CapX. As you can see, both capsids show comparable gene delivery efficacy, and both are able to lead to widespread CNS distribution. To summarize, the TfR1 capsids at Apertura have been engineered with accessibility in mind, and these modifications that confer resistance against neutralizing antibodies are compatible with both manufacturability as well as with their ability to target specific tissues of interest. These capsids have also been engineered to address other challenges, including intranslatability, selectivity, and manufacturability. In conclusion, IV-administered neutralizing antibody-evading AAV capsids can greatly expand patient access to gene therapies. IV administration is a procedure that is minimally invasive, it's not complex, and so it can be used to globally expand access to gene therapies, especially compared to intracranial administration. Antibody-evading AAV capsids can expand access to gene therapies by enabling more patients to be eligible to receive gene therapies and to enroll in clinical trials. And at Apertura, we have developed an assay that allows us to understand a capsid's ability to evade neutralizing antibodies. And furthermore, we have engineered capsids that have been able to evade these neutralizing antibodies while still preserving their manufacturability and their ability to reach tissues of interest. At Apertura, we believe that we're leaders in receptor-targeted approaches for capsid engineering, and we have used our platform to develop next-generation vectors. We are open to working with small and large pharma, academic laboratories, and foundations, and have flexible partnering opportunities, including licensing as well as co-development opportunities. So if you're interested in learning more about our capsids or platforms, we would love to talk to you. I actually have to transition to a new different session for my talk on manufacturing shortly, but my colleague Von Wiltman, CMC Program Manager, will remain here for the following Q&A session. Thank you all.
I'd like to introduce, for a woman that needs no introduction, Dr. Nicole Paulk. She is the founder and CEO of exciting new company at Siren, Biotechnologies, but she's had over 2 decades experience, as we all know, at UCSF in the AAV vector. And today, we're going to hear her work on a universal AAV approach.
All righty, Rue, let's get rolling here. So, again, my name is Nicole Paulk. If any of you don't know me from my past life as a professor of AAV gene therapy at UCSF, and I'm actually going to talk to you today about a project that actually started way back in 2017 when I first started my lab at UCSF, where kind of through this lens, very, very fortunate as a professor, you very often get asked to sit on a lot of scientific advisory boards. And so you kind of get to see the nitty-gritty underbelly of a field in those very early tender moments when a company is 2 people and an idea. They maybe not have even closed their round yet, and they have a cool technology, and they're like, how are we going to apply this? What diseases are we going to go after? Where should we target? How should we think about this? And so you kind of got to see what are the earliest challenges that, whether it's a teeny tiny startup getting started, what are they facing, and what are the challenges in the AAV gene therapy field, all the way up to the big publics whose SABs I sat on, where they're like, what are we thinking about for our 5th program? And kind of everything in between. And as a lab, we're kind of whiteboarding out what are the biggest, gnarliest challenges that the entire AAV gene therapy field is facing that probably might need to get solved from within academia, because it's just going to take too long for industry to really go after something that gnarly. And one of the challenges that we saw, that we kind of lovingly referred to in the lab, right? Every project needs a name. We lovingly referred to it as the Universal Payloads Project. And that was because one of the challenges that we saw, we got pointer here, one of the challenges we saw was that every single time, either our own lab, when we were queuing up a project and thinking through what kind of novel technologies to develop, to all the various companies that I would advise, one of the first questions that gets asked is like, oh gosh, maybe we have a new capsid, right? Maybe you got a capsid from Apertura. Maybe you have a new payload. Maybe you have a new manufacturing method. Maybe you have a new toggleable switch. Maybe you have a new whatever, something really cool. It's like, okay, well, now how are we going to apply it? Where are we going to point the ship? And how should we think about everything that's going to go into our drug product, right? Every one of the component parts. And what always would end up happening, 9 times out of 10, no matter what would happen, if you picked your rare disease you wanted to go after, no matter what, you had to change the payload, because you're always going to go after a different rare Mendelian monogenic disorder. And so the payload, the cDNA, right, the medicine, the juice that we're going to put in these things is going to change with every single indication. So no matter what would happen, we would take that first meeting and everyone would go, well, since we're going to change the payload, like the drug product's going to change. So we might as well, we're going to need we need the best capsid. And we need the best promoter, and we need the best regulatory element. We need to do a huge, CDMO manufacturing comparability study, and we're going to need to find new partners for all of these things. And all of a sudden, you've got 3 years of kind of discovery and dev work before you could really queue up your development candidate and actually go after the disease of interest with your new technology. And it all stemmed from the fact that no matter what, the payload was going to change every single time. And so that meant, well, let's just optimize everything. So we wanted to kind of throw that on its head and ask the question, well, what if we didn't change the payload? What if you had a single AAV where every single component part, including the payload, did not change? And so you could use a single AAV to treat 2, 3, 4, 500 different things. Could that even be possible? What could that universal payload be? And so started to take a kind of bioinformatic approach to that and thinking through what are payloads we could provide that would modulate things of interest across different disease spaces and tissues and developmental time points. Right? So things like transcription factors and cytokines and things that are very pleiotropic and involved in many different tissues and pathways throughout the body, and wanted to see if that could be possible. Could you develop a universal AAV where every single component part of that virus, including the payload, did not change, and then you could use that to treat a variety of different indications and now drastically reduce the amount of capital needs that you would need for every one of the programs, I think one of the themes we've heard at this meeting so far is, oh boy, it's getting a little hard to raise capital, isn't it? So what if you could reduce the capital needs per program, reduce the development times, both preclinical as well as clinical, so that you could start to crank out these indications still in the rare disease space much more quickly. So we decided that we would kind of blend two fields that we know well and that are well validated, right? So AAV gene therapy, but now with cytokine immunotherapies. Our favorite cytokines are interferons. I'm going to skip 7 years of work where we chose that, but suffice it to say, trying to come up with a new class of modalities, these AAV immuno-gene therapies, where now we can deliver cytokines with AAV gene therapies, and again, not change those payloads across the different indications that we're going after, in this case, cancers. And we can address, for the most part, all of the major challenges that the normal, like the protein delivering cytokine teams, have been facing for the last decade around challenges around durability and toxicity and lack of specificity, right? We can address all of these by vectorizing these and putting these inside an AAV gene therapy. We think the brain is actually a really great starting kind of landscape from which to start this process of testing AAV immunogene therapies. It's immune privileged. We can do local delivery with very exceptionally low doses. This is an established method that we've used now, up to and including FDA-approved products, right? So this is how PTC delivers their AAV for pediatric Parkinson's, right? AADC. There's established precedent for going after the brain. So we've got clinical collaborators and teams now from all of your fantastic companies who've gone into the brain. So we've got established precedent. Our clinical colleagues know how to work with local delivery into the brain with these nice low doses. And we've got broad applicability. So for the most part, we've never met a tumor we can't hit with AAV when you're delivering locally. And so we can really go after all 126 primary brain cancers, as well as the metastatic market, again, with just a single capsid, single drug product, single delivery route, single dose, single administration, single everything, and really just start going down that list of all 125 of those brain cancers. If we start at the very top of that list, which one is the most prevalent and most likely to go after? That's going to be the high-grade gliomas, right? So these are the most common brain cancer in adults and second most common in kids. Huge numbers, no effective therapies. I think we all know this space. We actually think that intratumoral delivery is going to be and should be the preferred route of administration specifically for the brain. So we're not talking about doing systemic administration and getting up into the brain, even though there are fantastic capsids that can do that. We think that intratumoral is actually going to be preferred for safety reasons, for all kinds of safety reasons. Happy to get into that in more detail after the session if folks want to come up. So using CED deliveries, this is a validated method for very targeted delivery where you can control within a 0.5 millimeter where your virus does or does not go, and you can pair that with MRI guidance to get even more precision so that you can make sure this never leaves the margin of the tumor in the brain. So let's start getting into the data, the fun stuff. So this is now data using Siren 101. So we've made some even kind of more fun edits compared to the things that we had back at UCSF. So this is now the Siren data using Siren 101, and we can demonstrate, I've got about 4 slides here on some of the actual fun data. And so the way the kind of slides are oriented, we've got a schematic on the left and then the data on the right. So in this case, we're talking about xenografts. This is CDX. So we take slightly immunodeficient mice. We can transplant them with human brain tumors, either in the CDX or PDX context. So CDX meaning basically immortalized lines, PDX meaning direct primary patient lines from tumors. In this case, what's on this slide is the CDX data. So we can transplant these mice, infuse them using the exact same method, CED, as we would in the clinic, and we can see very rapidly within days a 98% reduction in the tumor size of these animals relative to animals that receive either saline or an irrelevant AAV that just carries a GFP payload. So you've got to actually express these desired payloads. Just putting an AAV into the brain isn't going to do anything beneficial for a tumor. You actually have to express these cytokine payloads to see that tumor reduction benefit, and it's highly significant. If we look at that exact same experimental setup on the left, but now we're just looking at survival instead of tumor sizes, as you can imagine, if you can obliterate a tumor, that leads to improvements in survival that are quite significant. So we can see an 86x increase in survival in just 11 weeks in these animals. And that's all the small animal data. So now if we pivot over to large animals, what can we see? So this is actually like hot off the press data. This is the first time we're talking about it. So we just completed an exploratory kind of pilot study in large animals now. So pivoting over to pigs, and these are fully immunocompetent animals. So this is to address safety rather than efficacy. And we wanted to see Could we show first feasibility of our infusion method? So using those CED infusion rigs with MRI guidance into the brain of now a large animal, could we feasibly queue that up and be ready for the clinic? And also from a tolerability perspective, and now in a fully immunocompetent animal, looking to see what's the immune response and all of these types of things to these infusions of AAV when done locally with MRI-guided CED into the brain. And it worked fantastically. So all 6 of our exploratory study pigs had normal in-life observations, everything shown here. So body weights, clin chems, CBC, coag, you name it, clinical pathology reports, they were absolutely fine. They tolerated this fantastically. So this is kind of the combined body of data. We've got PDX, CDX data, syngeneic allograft data, now large animal data. I don't have time to go through all of these. We've got a really nice body of data to go in front of. We submit our pre-IND packet to the FDA next month, and should be able to hopefully complete our IND-enabling animal studies later this year. On the other fronts that we're going to need for that IND, for this first kind of use of a universal AAV gene therapy, is on the manufacturing side. And so as we announced last year, we're partnering with Catalent on this for both plasmids as well as vectors. Our GMP plasmid production is complete. The scale-up and process development at scale for the virus is complete, and we'll be doing the vialing and release of our GMP drug product later this year. So all in time for the IND. On the regulatory side, we announced just before JPM that we got both ODD and RPD late last year for both adult and pediatric gliomas using Siren 1.1, so very excited for that opportunity. And now we're really kind of ready to start demonstrating this platform for this first time. So high-grade gliomas in adults is where we're starting, but it's by no means the end. And so if you kind of start from the bottom and work your way up here, the intent, even though it's not popular at the moment, but I promise you the ball's swinging back. Platforms are going to come back. You are going to need to be a platform in like the 26, 27 era. So get ready for it. There's the swing back from assets is going to come back to platforms, I promise you. And that's where we're gearing to be. We're gearing to be a platform model. So the world's first universal AAV gene therapy that can go after multiple things. We're obviously going after cancers to start, but that's just the beginning. The whole point is that all that process development we did in 23 and 24 is done, and we don't have to do it again for any of our future programs, right? It's done. So we can now leverage that into each of our future programs and not have to repeat that time or that capital. We have, go back. We have the ability now, particularly if we stay in a single tissue, if we stay in the brain and we go after those 126 primary brain cancers and the metastatic brain cancer market as well. Right now, we have this fantastic ability to leverage all of the safety data from that first program into each of the subsequent programs because we have not changed the drug product, the tissue, the route of administration, the dose, the clinical trial designs, what the endpoints will be, any of this. So it can start to become cookie cutter so that we can do this much more quickly and go after every single one of those cancers in the brain, all of which are rare. So we're still in the rare disease community. It's still the heartbeat of our company, but now we're in the rare cancer space rather than Mendelian monogenic space. And so we want to make sure that we can start to get these to patients as quickly as possible so that we can improve access. And what's great is this scalable across cancers, because again, we don't have to change anything about the drug product, optimization, manufacturing, anything, make one lot of this and use it as much as we possibly can. The goal is to kind of become like the Keytruda, but of gene therapy. So with that, I hope you're as excited we are about switching over to becoming a clinical stage company in 26. And I don't think we're doing questions until the end, so au revoir.
So you can see already a lot of exciting talks about the promise. So next we have, I'd like to invite to the stage Dr. Kinnari Patel. She is currently the President and Chief Operating Officer at Rocket Pharmaceuticals. She oversees the regulatory, clinical execution, global program teams, alliance management, CMC, quality development organization. She has many, many years in research and R&D with regulatory science, policy, quality, compliance at leading pharmaceutical companies. She received dual degrees, a BS in biology and Doctor of Pharmacy. So today we're going to hear about her work with the next generation AAVs at Rocket. Dr. Patel?
Hi, everyone. It's going to be a little less science, a little more holistic, because I realize a lot of individuals here may not know about Rocket and the work we're doing, but I will focus on AAV pipeline. So we are a company focused on rare diseases and monogenic diseases, and to really provide treatment options for diseases that have no other treatment options available. We have a long history of being passionate about the science, the CMC, and I love being at this conference because I get to learn the new science age, and I get to learn about the lessons learned in collaboration. So that's really beautiful. So a little bit about our company. We are trying to develop first-in-class, best-in-class therapy, and that's really our mission. And how we do it, it's a little bit of video, which says it better than I could. So let me just play that for you.
Our DNA is the blueprint of who we are, shaping our destiny. But for about 240 million people around the world, a gene not working properly within their DNA could be causing a rare and potentially life-threatening disease. After decades of research from leading scientists and clinicians around the world, a genetic revolution is underway. The goal? Delivering working copies of the faulty gene to the body to correct the disease at its root cause. This groundbreaking area of research is called gene therapy and has achieved remarkable progress. In fact, there are currently over 30 gene therapies approved in certain countries around the world. Rocket Pharma is at the forefront of this genetic revolution as we strive to redefine the future of medicine. We are harnessing proven gene therapy technologies to develop potentially transformative treatments for patients living with genetic diseases. With the patients as our North Star and inspiration, our team is guided by 4 core values that are embedded within everything we do: trust, generosity, curiosity, and elevate. Rocket is powered by a team with decades of experience bringing therapies to patients. We foster a deep understanding of each disease in order to develop potential gene therapies for conditions where patients have limited or no treatment options. In close partnership with the patient, medical, and scientific communities, we strive to ensure patient access and will create the blueprints to develop treatments for more diseases. We are collaborating with the world's leading minds to develop and apply cutting-edge gene therapy science to rare bone marrow and cardiovascular diseases. We are relentless in pursuit of our mission to seek gene therapy cures for patients with rare and devastating diseases. Cures that may represent freedom for patients and their loved ones so that they may live without the burden of having a genetic disease. We are in pursuit of a world where DNA is no longer destiny and genetic diseases are history.
So DNA is no longer your destiny. And as you could imagine, this year we're heading to commercial stage. Hence, we have a beautiful video to go along with everything we're doing. How we started was really simple. We said, hey, there's 7,000 rare diseases out there. What are the diseases that have no treatment options? What are the diseases where there are actual technologies like AAV, like ex vivo lenti, and platform that's de-risked that we can apply in a way that can make these therapies available to patients today, now? And based on that, we ended up following down to patient populations and monogenic diseases that focus on 2 really platforms, ex vivo lenti and AAV. We ended up focusing really on kids because we realized the last 20 years, kids are an afterthought. And I know there are a lot of legislative things that have happened that to make pediatric rare diseases not an afterthought, but really forefront of innovation. So we focused on that, but we're not open. We're open to everything that's available as long as there's a significant unmet medical need. Unfortunately, what we realize in some of the videos, you see pictures of kids we've treated. We've been fortunate to treat amazing kids that are living a normal life and full life. And so DNA no longer being your destiny is really our North Star. With Rocket Pharmaceuticals, we're fully integrated. We realized a few years into the company that, hey, CMC, we have amazing CDMO partners, but if we are really going beyond 1 or 2 or 3 ultra-rare diseases, the control, the cost of goods, the quality, the platform of being able to do your own innovation is really critical. So we brought in manufacturing in-house in Cranberry, New Jersey, and moved our headquarters from Manhattan into New Jersey in order to have a 100+ thousand square feet, where even the basement is built out, which our scientists love. It's very New Yorky in New Jersey. But we can go from an idea of vector design all the way to 800-liter commercial grade manufacturing in-house for AAV. Our ex vivo lentis partner with a lot of amazing CDMOs around US and Europe. And I'm not going to go into this because all of you know what these technologies are all about. But what I will spend time on is a minute on talking about our ex vivo lenti pipeline. We have 3 products that are in equivalente. The data you'll see from phase 1, phase 2 that are publicly available are just amazing. When gene therapy works, it really works. Kresladi, which is under review with FDA, we're resolving our CRL comments for CMC analytical components. And that disease, 9 out of 9 patients, I'm a pharmacist, many of you are healthcare providers. I've never seen data where 100% of the patient had 100% efficacy reached in every single parameter. They're not just living past the age of 2, where 2/3 of them would die by the age of 2, but they're thriving and they're living a normal life and back in daycare. So to see them and how well they're doing, it is really beautiful. And it's one of the reasons we do what we do. Fanconi anemia is very unique because Fanconi, a lot of you guys know about this disease because it's something that's been researched for years. And we finally found a way that gene therapy works thanks to our partner CMAT at NIH equivalent of Spain in Madrid. And what they found is there's a selective advantage. So the disease biology is complicated. But what we found is with that conditioning, we can actually take patient cells, give it back, and over time, the corrected cells actually re-establish itself in the bone marrow. And we see these patients now living without a bone marrow transplant need or bone marrow failures. And we see their hemoglobins and all of these biomarkers over time increase. And that is a beautiful thing because unfortunately the FARF organization, which is Fanconi Anemia Research Foundation, renamed itself to say FCF, Fanconi Cancer Foundation, because a lot of the patients that had transplants before are now unfortunately passing away with secondary cancers. So this is something that we could potentially prevent with gene therapy. And our last program is PKD. PKD is very unique because a lot of our patients in phase 1 that were treated had hemoglobin of 6 or 7. As you guys know, normal range is 12 to 14. We've seen all 4 patients that were treated are not just transfusion independent, living a normal life, but their hemoglobin stabilized into the range of 12 to 14. And it's been like that for over 2 years, which is beautiful. So that's in a pivotal study. And Fanconi is under BLA review as well as MAA review with EMA. So let me focus on the topic for today. How do we make these diseases and how do we make the technology we have applicable to even larger diseases beyond ultra rare to just some common diseases? For Rocket, the proof of concept really came with Danon disease. Danon disease is very unique and it's unfortunately devastating. It's an X-linked dominant disease. What that means is, unfortunately, the boys that have Danon disease, they pass away uniformly by the age of 19 or 20. What we've seen is the females, quote unquote, live longer, but it's not longer. They pass away by the age of 35. So that is still devastating when you talk about cardiomyopathies and people living into the 60s and 70s and 80s before they have a need for transplant. So what we were able to show with our Danon program is when we replace the missing gene, we were able to see not only these patients surviving, and we had a kid, his mom sent us a Christmas card. He's 25 now without a transplant, living and having his own apartment and living his own life, which is beautiful. So this is the data that's really caught the attention of not just health authorities, patient advocacy, but also New England Journal of Medicine. We were able to present our phase 1 data. As you know, that's ultra rare. Less than 2% of the publications, I believe, make it to phase 1 data into New England Journal of Medicine. This data shows that when you replace a missing gene for 6 out of 6 evaluable patients, not only did we see LAMP2 expression in their heart when we did the heart biopsy, which was 0 negligible at baseline, but we were able to see changes that lent itself all the way from, pro-BNP and cardiovascular biomarkers like troponin, BNP, etc. But we were also able to see NYHA classification change from class 2, which is symptomatic, to class 1, asymptomatic. So all of these patients are asymptomatic. KCCQ, which is a quality of life measure, as you might know, regulatory approvals for KCCQ-based parameter has been a delta difference of 5 points. Here, mid-20s is the average point of difference. These patients went from not being able to just walk down their own stairs or go class to class to now living a normal life, going to college and having their own place to live, which is the reason we're really excited about this program. This program currently is in a phase 2 pivotal study agreed with the FDA, MHRA, as well as EMA. 12 patients to be treated. And what's beautiful about biomarker-driven accelerated approval pathway, which FDA absolutely understands, is this. We have to look at their patients at baseline, their biopsy, and then look at it at 12 months. And if we see a delta difference between no LAMP2 gene expression to LAMP2 gene expression, as well as LVMI change, then we can lead to an approval for an accelerated approval. these same patients being followed out to 5 years would lead to a full approval. Now, if you talk about drug development efficiency, to me, this is a great hallmark of drug development efficiency. We learned a lot during our phase 1 and phase 2 programs, which includes for safety, right? There were a lot of situations where a lot of companies have dealt with safety issues, TMAs, and et cetera. And there are amazing consortiums available. Dr. Barry Burns here, Dr. Barry Greenberg. They're amazing experts that we work with in order to work as a community with other biotechs to figure out how do we manage the safety? Because what we saw in phase 1 data for Dana and what we're seeing in even our phase 1 program for PKP2, initial safety challenges that you have or issues you see or side effects, they resolve within the first 2 to 4 months. But the efficacy you see is lifelong, or at least we believe it's lifelong. We've been able to show the durability out to 5 years. And because cardiovascular cells do not turn over, and essentially the cells you're born with are the cells you're going to have for life, the thinking and the scientific theory, which we'll hopefully find out years from now, is this is one and done curative therapy. The next program we use is PKP2. PKP2 is another devastating disease. Here we apply the lessons learned from Danon disease, and we're able to work to really establish what is the therapy that we want to develop. AAV9, R74, RH10, we did head-to-head studies preclinically, and we, for many reasons, decided to go with R74. One of the main reasons is this is a disease that really impacts adults. So if you're treating adults through an IV infusion, if we need to go to a higher dose, we wanted to make sure we leverage the experiences available from other companies that show you could go to a higher dose and do it safely. So to allow that flexibility, we ended up switching our platform to R74 for this disease. And this program is currently under our phase 1, there's preclinical studies that are published and available. This program is currently under phase 1 study. We did announce recently that our cohort 1 has completed enrollment and treatment, and we hope to have that data available this year. And it doesn't end there. When you can actually do it once, you can potentially do it twice. Why not keep going, right? We realize that patient communities really need therapy and treatment options for heart diseases. So the next IND that we have been filed first half of this year is BAC3. So BAC3 DCM is very interesting. With this, we'll have HCM and DCM covered. We'll have different platforms that are going to hopefully show the benefit risk and the synergy of platform. And our goal is not just to use this data to help these therapies come available to patients more efficiently as possible, but use this knowledge to really target diseases that are even bigger. Monogenic disease is where we're focused. If you combine these 3 diseases alone, within US and Europe, you could treat up to 100 plus 200,000 patients across. So when you're talking about 200,000 patients impacted, that's no longer rare, right? By definition, it's greater than 200,000, no longer rare. But if you take this technology and apply it to more monogenic, or maybe using some advanced science that's available, polygenic and multigenic diseases, I think honestly we could change the humanity, and DNA truly would become no longer your destiny. And that's the dream we work on at Rocket. So how do we get started, and where do we get started? Well, unfortunately, what you realize is February is coming up, and February is a rare disease month, right? This is when Orphan Drug Act in 1983 was passed by Abby Myers, who was a mom struggling in Connecticut for her kid to have access to therapy. Even, 40 years later, we're still struggling. And the struggles when we talk to patients is genetic testing. If we're targeting pediatric diseases, if we're targeting diseases that are not reversible because of the genetic mutation or this missing gene, then waiting till somebody's 20, 30, 40 might be too late to reverse all of the manifestation of the disease. So how do we get patients treated as early as possible? What's really important is actually to have genetic testing available. So we focused a lot of our resources in the recent years on that. And I'm happy to announce that the benefits of genetic testing, it's not just about insurance companies saying, hey, we can make it available if you want for a few $100s, or it's covered by insurance. What do you do once you have those results? So genetic counselor, the ecosystem is still quite complicated. So we're working with institutions across the US and Europe to figure out how do we connect patients that may think they have cardiomyopathy or genetic disease. How do we get them tested? How do we get them the counseling and the support needed to get the right diagnosis? Because if we get the right diagnosis, we may actually be able to save a life before it's too late. So we've partnered, we realized we can't do this as a small biotech ourselves. We partner with many individuals and organizations, including Vitae and others in Ready. And I'm happy to announce that in November of last year, a few months ago, we launched Mission Genome. Mission Genome is a way for patients to reach out anywhere to get free genetic testing and free counseling. And have their KOLs or the primary care physicians actually be able to support how do you get diagnosis and the correct diagnosis to figure out, are there treatments available, clinical, commercial, etc. So we're really excited about that. And this is the beginning. I think we're in the amazing space where 2017 was one of the first times that, Kymriah and Avexis and Spark had their approvals, and we've come a long way on one hand. I think we have about 20 or 30 drugs approved, but it's not far enough. So I think the work you guys are all doing together, if we can collaborate, share our science and innovations and what worked as well as what didn't work in each of our cases, I think we can get there. And this is just the beginning. For Rocket, we have 6 pipeline and more that are undisclosed, including one other therapeutic area. So one at a time, when we get a drug approved, a new one goes to an IND, and that's our hope, 1 new IND a year. And at the end, I do want to take a minute to thank the patients. Honestly, this wouldn't be possible if patients and rare disease parents and advocates weren't trying to get more therapies approved. For us, every star on our wall, as a publicly traded company, I realize when our bankers and investors come, I do need to make sure that the stars are only there that are publicly disclosed, number of patients, but we do have a wall of stars where all of our patients that are publicly disclosed numbers, they're all up there. And it's really cool because each Rocket team member that had the most hand in making that happen get to actually hang that star. Empire State Building is where our original headquarters was, and we still have offices there. So we worked with Empire State Building as well as about 100 plus monuments around the world to get Rare Disease Day lit up. And what we've done is, this amazing, all corners of the world are trying to have these beautiful lights on February 28th lit up, because when a rare disease patient and their family struggle, they may not have a treatment being available. They may not know where to go. But that one night, we want to give them hope that we're thinking about you. And there's a whole community of scientists, sponsors, academics, everyone that's working really hard to bring them hope and hopefully bring them treatment options. So if you've not participated in your own companies or organizations, you could just do a simple thing is light up your own buildings, and it raises awareness on rare diseases. So with that, thank you so much for being here, and thank you for the panel opportunity.
So we will have a Q&A with our panelists, but before we get to that, I would like to invite Manny up to the stage. Manny Otero is the Chief Technical Officer at Lexical Therapeutics. Previously was the Chief Technical Officer at Origin Biotherapeutics, and he was till there. But basically, I want him up here because I wanted to ask him questions first before I open it up to the rest of you. Okay. He's working.
Thank you, Audrey.
Thank you. Yeah. Thank you for willing to be sitting here in front of me and asking these questions. So we've heard a lot. Gene therapy is really historically being focused on sort of small, rare disease, ultra, and as Dr. Patel said, the real thing is we want to move for larger therapeutics. So maybe you can tell us what you're doing at your company to strive for that.
Yeah, I'd be happy to thank you for having me here. And I think it's a wonderful lead in to go after that presentation. So much synergy with what we're doing at Lexeo. So Lexeo was founded in 2021 with the idea being that gene therapy was going to play a role in larger disease indications. We have an Alzheimer's program, and then we also have built a cardiomyopathy pipeline. We have a PKP2 program similar to what I just heard about. And then we also have a program in Friedreich ataxias. And right from the very beginning, the company, especially from a CMC perspective, said, we need to build a platform, a capability, a team that is going to deliver tens of thousands, hundreds of thousands, potentially millions of doses, global distribution. How do we do that? And so my background, so I spent 13 years at Merck Pharmaceutical Company, all in vaccines, R&D, vaccines manufacturing. And when I first got into the gene therapy space, it was very... it was eye-opening because, of course, people would tell me about how novel it was to make large doses of live virus drug product. And, of course, I come from a world, so I supported the measles, mumps, rubella vaccine, as well as ProQuad, which is measles, mumps, rubella, and Varivax, pediatric chickenpox. We are used to making millions of doses per year, global distribution, well over 100 different jurisdictions around the world, multiple analytical labs around the world, doing analytical comparability. I know this is a topic close to your heart. And so there were a lot of lessons learned from that. And so at Lexeo, very early on, which I suspect, I wasn't there for that, I suspect was unpopular. We elected to invest in an SF9 insect cell culture baculovirus manufacturing system. We also went ahead and built a CMC development lab as part of our headquarters in New York. And so we have a full drug substance, drug product. We're able to do process development, analytical development, formulation, all within our own control, and then set up key partnerships with key vendors, key CDMOs, and we have a scaled down model for our process.
No, that's great. I mean, as something I've always said for having the doing your analytical work closer to the floor, to the manufacturing floor, and you've heard a lot about that. I mean, so the fact that your company has strategically done that, it creates sort of an ecosystem so that your scientists and quality can work together. And of course, the advantage of just taking the sample down the hall, right?
Yeah, that's exactly right, Audrey. And it's two categories of analytical development. You have what I would call sort of a standard release testing, characterization testing, assays that we're all familiar with these various platforms and so forth. But as we all know, and you progress from early stage clinical development to late stage commercial, there's now expectations around mechanism of action, potency, assay development, having a mature stability program. And so we've actually invested in that from the very beginning in-house with novel potency methods for our products that, of course, have to link to clinical data and attributes of the product to clinical performance.
Very good. I'm glad to hear that, about putting that, what quality by design, or at least having that idea that even the early R&D assays might end up being your CMC lot release assays. So we've heard a lot of talks today and in 2 days about, making sure you keep the end in mind, the goal that we want to move forward to. And you mentioned earlier your viral vaccine background. It's almost 180 degrees difference in terms of approach, with the manufacturing and being able to deliver tens of thousands of doses. Do you see us moving there someday with lessons from the viral vaccine manufacturing?
Yeah, there's no question about it. Some of the lessons and themes that I see is that first of all, processes are going to evolve. We often find ourselves in the world of comparability. And so what was suitable for phase 1-2 development may not be suitable for phase 3. I think comparability, thinking about a comparability protocol in that framework right from the beginning is critical because you're going to need those improvements. That's one step. And the other step too is to really challenge, convection. I recently saw a talk by Peter Marks over at CBER, and he was talking about the AAV platform and trying to have a standard template approach. And he was talking about an anchorage-dependent process. And I've had a chance to connect with him, and I made the comment that this is probably going to be suitable for some subset of developers, but this is not where the field is going. If we think about the number of doses, the scale we need to reach, ideally we're going to leverage technologies that not only is about scale, but it's about robustness, it's about consistency, it's about the lot-to-lot variability. These are all the things that in a live virus vaccine context, we have been working on for the better part of 30 years. And so I think there's a lot of lessons to be leveraged there.
That is a great note to end my question. So I like to invite the other panelists up here. And Vaughn, can you come up as well to substitute for Jorge? And we could happy to open questions for the audience. I will walk around with the microphone like Vanna and see if there's any questions.
Just a question with regards, Nicole, to the gliomas. In clinical trials, from what I understand, normally like the standard of care is you have to resect the tumors. How do you guys get around that?
You can go after, the beauty of doing intratumoral delivery is that you can go after either resected or non-resected tumors, so you can address either population.
Topic is promise of future gene therapy. I know you all sleep with the ideas in mind. Any other questions?
Hi, I'm Elizabeth. Perhaps this is a little in the weeds, Nicole, but I guess in your model, you have really great data. Have you how much have you stress tested that? How big can the tumor get where your therapy is still successful?
So we haven't announced yet what the tumor size cutoffs will be for the trial, but we are testing a variety of different sizes to see how big we could go and how small we can go as well. So Stay tuned.
Sounds good.
So this one's for Lexeo. So would love to get your take on, you're using SF9. How have you found that compared to, a lot of other people using other platforms? How have you found it compares in terms of manufacturability and quality?
Yeah, thank you. So a couple notes there. So I think we've spent a lot of time thinking about yield titer productivity, obviously, and making sure that the platform could deliver there. And it is really a platform technology. So a note to this idea that platforms are going to be back in vogue in time. What we found is a few things. So first of all, the process for SF9, I think you often hear out there stories that was effective at relatively smaller scale, but as they scaled up beyond 200 liters, they had challenges. We've taken that head on. We have a small scale model system where we've been able to do custom media development, custom process development. We found that to be really critical. We've got yields coming out that are far in excess of 1E15 VG per mil, fully purified. We've demonstrated that at 200-liter scale in GMP. And as indications come along the pipeline, that requirement, we've got confidence that we can go higher there. Obviously, the advantage, or one of the advantages to SF9, is that you don't necessarily have the plasmids and other inputs that can certainly be an added complexity, added COGS, and so forth. So we think that we've got a system that we can work on for the future and is going to deliver COGS that are much more in line with where we think this needs to be. And to be specific to that, we need to get to a place where we're at thousands of dollars per dose and hopefully lower than that. To give you some context, when I was responsible for global lifecycle management of the MMR2 vaccine, this is 3 unique live viruses in measles, mumps, rubella, drug substances mixed into a single drug product, sterile aseptic manufacturing. This had a cost of goods less than $6 a dose. So I don't buy it when people tell our field we can't get there or it's going to require some radical innovation. I don't think that's true. I think we've got a lot of additional both engineering and biology opportunities to get there.
Hi, this is a question from Nicole. This is Carrie Wong from KSQ Therapeutics. I'm wondering, with your approach, is there any thoughts of going outside of the brain, like to other cold tumor microenvironments?
We have a not so secret discovery team working on exactly that. So stay tuned.
Yeah, this question is for Apertura from Jorge talk. I'm from Repligen, Rachel Eggman. So I saw in one of his slide that even though you're working on the new capsid, the next generation capsid, and it's very specific because you're attaching the receptor, why are you using high dose in injection instead of, I would expect to have low dose?
Right. Yeah. So that, I think that the image in question, I don't have that in front of me right now. So I'm a little careful to answer that, but I believe that is some from a first generation of capsid that we've used more to illustrate the transduction relative to AAV9, not to pressure test the kind of like dose levels. We're currently working on some studies right now that are really kind of going down into the much lower doses, into the 1E12 kind of range. So yeah, that's the rationale there.
Hi, Manny. Mark Backer with
Hey, Mark.
Interesting to see you're on the insect cell, and I know the pioneers sometimes prosper; they always suffer. This is a sort of redevelopment of insect cell, I guess. Can you comment, I guess, about being on the less beaten path? Do you have to educate the vendors?
Yeah, that's great. Great point, Mark, and great to see you as always in the community. Maybe a few comments here. So no question about it. Insect cell culture baculovirus has a fairly bad reputation. I think it's fair to say, or at least it's not a popular one. It's ironic because, of course, this platform first made a splash, or one of the first examples was from the vaccine world. This is how GSK manufactures their human papillomavirus vaccine, where they had reasonable success there. It's also been used in other companies that are out there. So typically the first thing we do is we walk into a CDMO relationship, and the first thing they want to do is tell us all about their stable cell lines, their HEC-293 technology, everything they've done. And of course, we let them go through that process, and then we let the data speak. We share the data that we have, and at least to date, nobody has said that they can do better. And so the data has been convincing. We've been able to have great regulatory interactions across multiple IND programs at this point. We've got a path to scalability. I do think one thing that we are doing maybe a little bit differently. So CMC folks, you guys will know 5 years ago, a lot of us were in a position where capital was readily available. It was a very common use of proceeds story to build an in-house GMP manufacturing facility. In some cases, that made sense. In some cases, I think people are revisiting those decisions. We have never chosen that. And the main reason is because we think it's really critical that, one, you have a process fully defined. So the idea is not to build a beautiful, shiny facility that you then wedge a process into. It's to have a process well-defined, and you build a facility around that process. And two, we think it's going to be really important to have multiple products to share the overhead and the complexities of actually operating that process. So that thinking forces us to have really deep partnerships with CDMOs, and that's how we approach it. We approach it both large CDMOs as well as very boutique, smaller CDMOs. We try to diversify the supply chain, and we try to choose people that have that openness to really partnering with us on the SF9 insect cell culture. And so far, we've had good reception on that front. Yeah, thanks, Mark.
All right. Well, I want to thank you all for coming, making this interactive. Appreciate all the questions, and hope the rest, tell everybody this was the best session that you've ever been at.
Thanks, Audrey.